Bismillahirrahmanirrahim. We start another very important video on plan drawings for paper 3, which many of you have been asking me to do a video on that, and this is just a video on how to do a plan drawing, and we'll be discussing many different questions and what is the possible questions that can come in this year's exam and the future exams. The syllabus has mentioned that what are you supposed to be knowing the plan drawing of. So describe the distribution of the gas exchange system of the cartilage, epithelium, goblet cells, squamous epithelium, alveoli, smooth muscle. Recognize cartilage, ciliated epithelium, goblet cells, squamous epithelium of alveoli, smooth muscle, and capillaries in microscopic slides, photomicrographs, and electron micrographs. Then the important thing is recognize trachea, bronchi, bronchioles, and alveoli in microscope slides, photomicrographs, and electron micrographs, and make plan drawings of transverse sections of the wall of the trachea and bronchus. So plan drawing can only come of the trachea and the bronchus. Then the next one which can come is plan drawings of stem, roots, and leaves. This is again mentioned here in the syllabus. I'm showing you the syllabus and you must know the syllabus. Describe the distribution of xylem and phloem and transverse section of stem, root, and leaves of herbaceous dicot plants. Draw and label xylem vessel element, phloem sieve tube, and companion cell from microscope slides, photomicrographs, and electron micrographs. So, what do we have? We have trachea and bronchus, we have stem, root, and leaf, and the third one is plan drawings of arteries, and veins. In what? The important thing is TS, which means if this is a tube, artery is a tube, so transverse section means like this. But then it said also LS. LS means something like this, lengthwise, longitudinal section. So you could be given anything. I remember one paper 3 in which this was asked. So longitudinal section. So this is what you have to understand is these are the three things for which you must know the plan drawings. Now you can see this is the trachea. So you can see the trachea, what will be the lines be? I'm afraid I cannot do very clear lines because it is I'm just going to do a short one because I've got very thick. So you'd have to do this, but then you'll have to do 2 lines of this to show that this is what you're showing, the ciliated epithelium. Then this is the trachea. I've completed this for you. So this is the trachea, which is a C-shaped trachea. And of course, the muscle would be somewhere here. The muscle would be here, but of course, you can't see much of the muscle here. It's not very clear, but the muscle would be somewhere here, the smooth muscle. So 2 lines for this. And I'm just drawing it just to show you the 2 lines. And then you would draw the outline like this. Whatever is given in your paper, whatever sort of diagram is given in your paper, you would draw the outline of this. So this is what would be a plan drawing. You would draw this structure. You can draw this structure a bit. So now you realize if you remove this, what are you going to see? The plan drawing. A diagram like this, and then I'm going to remove it and show you what's going to look like the plan drawing. So this is what it would look like at the plan drawing. I have removed the slide. So in a way, you see how I did it. I just traced it out. But it shouldn't be, it should be bigger than the diagram given to you. I've just traced out the actual picture, actual photomicrograph. But I want you to draw a bigger one of it in the paper because the slide is going to be smaller. So you're going to draw a bigger version of it. So this is the plan drawing of a trachea. Now, if you look at another diagram which I've drawn from a paper which was given in one of the paper 3s. This was given in a paper 3, which came in a very old paper. I don't remember the year now, but this was given in an old paper 3, and this was given in the mark scheme of how you were supposed to draw this. And this was the plan drawing of that diagram, which was then given in that exam. Yes, I just looked it up, and this is the November 2008 question paper 3-1, in which you can see the picture of this trachea and also the plan drawing of this trachea. Here you can see another slide of, and it's a labeled slide, so I want you to just look at it as the wall of the trachea, and I want to read all the labels, and I want you to go through them, just a quick revision. Now, another thing about the bronchus which you must remember is that they have plates of cartilage. The trachea has a total C-shaped cartilage, but the bronchus has plates of cartilage. And you can see in this diagram, you can see it very clearly how there are these plates of cartilage. I'll just color them red. You see this plate of cartilage here, this plate of cartilage here. This, of course, could come, a question could come in your, even in your paper 2. And you can see this plate of cartilage here. So if this sort of a diagram comes and you draw accordingly what you see. So this is the discerning factor which tells you, and then of course you would draw the 2 lines for the epithelium lining it. This is the 2 lines. I'm sorry, I can't draw it very correctly. So this is what you have to see in a bronchus. You would see plates of cartilage. It's also mentioned in your book. If you just look at your book, the blue book, it has it in the book as well. Now we come to arteries and veins. Now in an artery, as you can see here, there's number one, you must draw the crinkled endothelium. So you must draw this wavy line like this. It's very important that you draw this like this all over around the place. And then you see there is this line which is the tunica media, which is made up of the smooth muscle. And proportion wise, it must be correct. So basically just 3 lines. So basically just 3 lines. So this is an artery as well. And this is the vein. Now you can see the vein doesn't have a crinkled endothelium, and you must draw it in the same shape. Why does it? Because you see when you make a slide, it gets squashed and it doesn't give that round thing. It just gets slightly folded, just like you press something and it goes out of shape because it has a thin wall. So you can see artery and vein. The lumen is very large, the crinkled endothelium in the artery, and there is no distinct tunica media. There's not much of a muscle in the vein, muscle in the wall of the vein. So this is what you would have to draw if I gave you an plan drawing of an artery and a vein. Another artery that you can see. So you can see this crinkled endothelium, which you must draw like this. And then this is the wall of the muscle. This will go all around. Please draw it exactly the shape that you see it in, and draw it bigger. I'm of course just tracing it on it, but this is for you all to understand that sort of you just have to trace it out. Some of you find it very difficult, and I really find it interesting to see how you all struggle with it, but finally you do get it in the end. Another artery and a vein. Now if you remember, this is the muscle layer. This is the muscle layer. Now you have to remember there are muscular arteries and elastic arteries. Muscular arteries are those which are near when they start entering the organ. Like the renal artery, as it enters the kidney, will have more muscle in it so that it can control or sort of change the volume entering per unit time, so it can change the pressure. So you can see now, of course, the outer wall would be something like this. And then there is this layer, some sort of a layer like this around it. And then the vein you would draw as you see it. Please, no broken lines. I might have made a mistake in that. So you have to see is that you draw it exactly like what you see it as. So 2 lines in a vein and 3 or 4 lines in an artery if you see some distinct structures which are different. coming to the plant ones. So we have a root. Now in a root, of course, you have to draw this as a double line, then this line, this line, this as a double line outside, and then of course this line. And why would this be a double line? Because this is a double line for the endodermis. And then of course you have a this is not very distinct, so you'll have to draw, this is the sort of area which has the xylem in it. You don't draw individual tissues, you just draw the xylem tissue. And then this of course all is the, you can draw some sort of a structure like this which is demarcating it, and that's the phloem, or you need not even draw that. So two lines here, then a third line here if there is a demarcating tissue, and then two lines here for the endodermis and then for the vascular. Now the important thing is the ratio. How much is the central thing? Is it 1/3? No, when you look at it, this is one area and this is one area and this is a little less than 1/3. So if this whole thing is 6 centimeter, how much is the central part? I say it would be about 1.5. It isn't exactly 2 centimeter. So the proportion must be correct. Now some of you draw a diagram like this and you draw the xylem here this small. So this would be wrong. So you'd have to be very clear. If you've drawn this line, then how big is the central part? You can see there's a lot of difference between this and this. Now this one would be wrong. This one would be correct, yes. Because you have done the correct proportion as compared to the whole diameter of the slide. Now this is a stem. In a stem you can see what we'll have to draw is, you'll have to draw two lines here. I'm not drawing it exactly, but I meant is this and this. And then you have to be very clear about how you're going to draw the vascular bundles. Now you can see the vascular bundles are not the same shape. They are slightly different. Some are smaller, some are larger. And you draw all of them very symmetrically. And you draw them all like this. But they're not like that. Some are smaller. There are one or two, three smaller ones. And then the very tiny one as well. And then there is a bigger one. And then there is this. And then there is this. And then there is this. And you can draw another line here because showing the sort of the tissue which is changing. The rest, please remember, there are no cells. So you would have to, this shouldn't be like this. There shouldn't be any broken lines. So you would have to, I would sort of draw it like this and this like this. Now, if it's a draw half, then you see in a half, you can either have, if you're drawing this half, then it could be 1, 2, 3, 4, 5, 6, 7, 8, 9 vascular bundles. But in this half, there are 5 vascular bundles. So, of course, in the mark scheme, of course, it will be given to us as examiners that anything between 5 and 9 vascular bundles would be fine. So you just can't draw randomly. You can't just say, okay, we'll just draw like this, just randomly as you like. You must count the number of vascular bundles, and in one half of a slide, how many vascular bundles are present. So in a stem, what do we have? One line, two line, a third line, and then the vascular bundles of different shapes. And you must also do the differentiation. You must see that there is the xylem and the phloem. So you must separate there is a line which is separating it, like this here and this here. So you must draw the vascular. I'm going to show you another diagram of the vascular bundle in detail. So the vascular bundle must have lines showing the xylem and the phloem and the area which is called the cambium. Now this is a square stem. So if I ask you to draw half of it, then you'll draw it like this. You'll just trace it like this as you see it. And then there's this line again here, a double line here. And then you see this is a vascular bundle, a huge one. And then there is one here. And then there's one something here. And then there's one here. And then there is some sort of a tissue here which is different. And here there's slightly different tissue. So half of it, now if you remove it, this is the plan drawing that you should make. Now this is a very detailed drawing of the central part of the root. Now if you see here, and if I had to demarcate it, you see this is all the phloem. The phloem is all this area. And then you can see this is the phloem area. This is the phloem which is like this area, like this area. And this area is the phloem area. And this area is the phloem area. Now you can see the labels here. You can see the vessel elements. Then you can see the pericycle, the endodermis. If you need to revise this, please revise this. The endodermis and the pericycle. A very good diagram showing you all the details. Now this is a detail which was required in certain situations in which they asked you to draw a high power drawing of phloem as seen in transverse section. This is from your book. Now as you can see here, now if you see this area, wherever two vessels meet, there are three lines. So there has to be three lines where they meet. And two lines here, you can see here, this area has two lines only. This area has two lines only. This area has three lines. So wherever there is these three lines, you can see here three lines. So wherever there is this, you have to draw three lines. I'll just show you another diagram as well. Again, a high power drawing of the xylem vessel again from your book. You can see three lines here, three lines here, two lines here, two lines here. Because the xylem is shown as a double line because the xylem vessel is thick because of the lignin deposited in it. Now you can see this was the drawing which was given for your from your book. We have this detailed a leaf because this is the one which I want to talk about, a leaf. This is a transverse section of a leaf. Now if you look at what is the plan drawing like, the plan drawing was like this. This is the plan drawing. This is the plan drawing of it. So you can see there are just two lines to show the upper epidermis, then the line to show the palisade mesophyll, and then a double line to show the lower epidermis. And then of course you can see here this is the central midrib which is shown. And then there is this mark here for the separating the tissue. So the first two lines are for the upper epidermis. This line shows where the palisade mesophyll is, and this line shows where the lower epidermis is. So very easy. If you have been asked to draw the midrib, then you would draw the midrib of the leaf. I'm just going to show you some more plan drawings of different leaves as well. You can see this is again from your book, the microtears of a young ranuncular stem. This is the top one, this is the stem one. And then this is the of a root. And then how you would be drawing this of the root. Please everybody have a look at it and understand it. This is the slide and this is the plan drawing. This is the slide and this is the plan drawing. This is from the blue book that we have, which is the standard book that we use to teach you all. Now, this is not a plan drawing, but this is a diagram of, sometimes they give you an annotated drawing, and this is a xerophyte. And I wanted to know the annotations on it, the hairs, the position of the stomata, the vascular bundle. The trapped humid air lowers the water potential gradient so that less water is lost by the stomata. Then hinge cells have shrunk due to dehydration. This pulls the sides of the groove together, and this is a waxy cuticle. So curl leaf of a xerophyte. You can easily see this is a stem because the vascular bundles are all on the periphery. So this is the stem, and you must know the labeling of it, the epidermis and the cortex and the pith, which is the central area, which is called the pith. Now you can see the labeling of a vascular bundle. You see, if you look at it, and some of you really find it difficult to understand this. This would be the vascular bundle. Now the phloem is here, this part. The cambium is this part, and then this is the xylem. So the xylem, the cambium, and the phloem. You can always Google these and see more of these to get them to understand them. I know you all say, you keep on thinking that Dr. Sarah always says Google it, but I mean, I think that is something which is all available to you all, and you all should really look into it. So the xylem, phloem, and the cambium. So you must differentiate when you draw a vascular bundle. You must always differentiate. You see, the phloem is always a thinner area, xylem, and then the cambium is a sort of a central area. The xylem is always slightly bigger. The phloem and the cambium is sort of a middle area. This is another leaf which has sunken stomata. And here, of course, you would see that there would be two lines for the cuticle, and then it's a three-layered upper epidermis. So this is the three-layered upper epidermis. But you won't draw it three-layered, you would just show it, you maybe annotate it and say three-layered upper epidermis, and then this is the vascular, this would be the palisade. The palisade is thick and is many layered, and then you can see these vascular bundles. This is of course a vascular bundle cut longitudinally. And then you see this area, which is again a three-layered lower epidermis. And this has of course three layers, but then you don't show the three layers, you just show it as a whole, that this is the lower epidermis. And then this of course goes like this. And then you can see there are gaps, so those are the stomata. So you would show this as a gap. You see, you'll have to draw what you see. And then of course you can see this thing here, which is a pretty big vascular bundle. And then if they say, okay, draw three adjacent cells of the upper epidermis. So I mean, where am I going to look at it? I'm going to look at these three cells. And these three cells are not very uniform. So I would do something like this. And then I would draw a double line here. Well, I of course do it in a very different way than you all do. I first do the double line all over, and then I just do three lines where they meet. Well, everybody has their own way of doing stuff. So this is where I would say that you'd say draw three cells of the upper epidermis. Any three. This slide would be much more clearer than this one, of course. And please remember, this won't be these broken lines which I might have done with the marker. This is another very favorite leaf which has come in many papers. And if you look at the boundary of it, the boundary of it would be something like this. If I ask you to draw a plan drawing of this, now here there's a stomata. So you can see all the labeling here. I'm not looking at the labeling and I'm not telling you. So there would be a double line here. This would be the line which is going to demarcate the epidermis. And then this line will demarcate the palisade. and then this area will market the vascular bundle. So you'll draw it all the way through. You see, because you'll show the epidermis as a double line, and then you see it become thinner here. So I hope this is going to help you, and please do go through all the different leaves and go through all the previous papers on this. There have been a lot of, please look at questions which have come on arteries, veins, trachea, bronchus, and leaf, stem, and root. There are many paper threes on this, and if you just type in leaf uh, slide 9700 paper 3, I'm sure you can find all the different papers on this.